now we'll have the closing presentation and we'll take a more in-depth look into his work and research around reptiles as models for differential evolution. And similar to the other presentations, we will have around a 10-minute Q&A session at the end. So Raul, feel free to take it from here.
Thank you. Thank you so much. First of all, thank you for inviting me today, and thank you for organizing this. I think it's a wonderful organization, and being able to talk about these topics that I think are relevant, and I think raising awareness of how reptiles are important is a very cool thing. So thanks for that. And today I'm going to share with you some of the research that I do and some of the research I'm passionate about, that uses lizards as models for study evolution in nature. So I would like to begin talking about what is evolution and what do we understand for evolution. So the first thing is evolution is basically the change of populations through time after every generation, and is associated with the genetic inheritance of the different organisms. And it comes from this idea that all life on Earth come from a common ancestor. So for instance, if we think of this all vertebrates, which are super different as we see them today, they all come from a common ancestral tetrapod. So that's the idea behind this. The cool thing about evolution, and when we look at the biodiversity patterns in nature, is that it's not equally distributed. So we have uneven patterns of biodiversity in nature. So this is an example from mammals. This is a phylogenetic tree showing the different kinds of mammals that we have in nature. And you can think that some group of mammals, like elephants, are a very few species that exist today. It's a very reduced biodiversity. But on the other hand, over 1/3 of the current mammal species that exist in nature are rodents. So that's very interesting. If you think of bats, they are in between the number of species of mammals and rodents. So what's going on here? Why are rodents so diverse, but elephants so species poor? There's something going on. And I approach this kind of question and this kind of problem using lizards as a model. And I focus on New World Iwanians or pleurodonts, which is just a small fraction of the biodiversity of reptiles. So this tree is showing the diversity of squamates that includes lizards and snakes. And this is only a small group within these species, but it includes some of the most beautiful and striking types of lizards. So iguanas, spiny lizards, and owls that I'm sure you're familiar with or have seen before. So why do I work with these guys? Because they have some of the most diverse vertebrate groups. So they have these 3 genera that are colored here that distribute in the Americas that have the highest or the biggest species number that we have in vertebrates. So for instance, anoles that are in the tropical Americas have over 400 species. So the Leolemys lizards that are from South America have almost 300 species, and Scalopus around over 100 species. So that's a lot of diversity. But on the other hand, we have some groups that are really species poor. So for instance, if we think of the Polychrus lizards that are sister to anoles, only have 8 species. So there is a differential way of accumulating diversity in these groups. So in that context, the question is, what could explain this uneven diversity? So that's what we need to understand, right? So let's go one step back and ask ourselves what promoters or what generates diversity. So first thing we need to know is that the process of speciation, it's what's creating new species. And the accumulation of differentiation of populations over time is what leads to speciation. So what kind of things generate differentiation. Of course, we can think of time, because at times, as time goes by, populations can differentiate. And also the degree of physical isolation between populations. So let's explore this a little more. Like, let's imagine we have a population of different kinds of lizards. So you can see we have a lot of diversity in terms of size and color. And let's imagine that we have a landscape change. So a river goes and separates these two that was one population into two different populations. Perfect. But now, just because of random processes and the random reproduction of individuals within each population, we will see that the frequency of the different variants is going to change. So let's say here in this population, the orange and the blue lizards are increasing in frequency, but on the other side in this population, the green and white lizards are increasing their frequency. So at the end, what we will have is that the populations have completely differentiated, and this is just because of the random reproduction of individuals after the two populations were isolated. Perfect. So physical isolation is important. And the other factor that could influence differentiation are the effects of natural selection. So let's explore this. So let's go back to our original population. And again, if we have an environmental change, let's say now the environment in which these lizards are living turns blue. And then the lizards that have warmer colors, like the orange and the yellow lizards, will be more easily seen by predators. Therefore, they're not going to survive as much as the other lizards. So we will have a change in the population, and in the next generation, the frequency of the orange and yellow lizards will be much lower. So here, because of the differential survival and reproduction of the individuals caused by the change in environmental conditions, this will be an effect on selection that is changing our population and is producing evolution. Nice. So that's the theory behind. So let's see if we can actually study and experience that with natural populations. So the first study that I'm going to show you is working with Leo Lemons lizards from South America. I'm originally from Chile, so I have studied these lizards for a long time. And the question here is, how do lizards respond to environmental variation? And we work with this species. It's called the dark lizard. And it's a tiny species, as you can see, like that's my hand. So it's a very tiny animal. They are super cute. And they live across very contrasting environments. So this species, so this is South America. And here we have the Atacama Desert, which is the driest desert in the world. And this species lives in the southern limit of the Atacama Desert, and they go south and live also in the Mediterranean forest of central Chile. So these two very contrasting environments, and these lizards are there. Just so you believe me, these are photos of both environments. So you can see here in the Atacama Desert, this is a very dry kind of landscape. And here we have the Mediterranean forest, so like very dense vegetation compared to what we have here. So we studied those lizards and tried to see if they were responding to the differences in their environments. So the first thing we found that they were very different genetically. This is analyzing the genetics of the different populations. And here you can imagine that in this plot, we have a lot of like tiny columns, and the color of each column represents the probability of it belonging to a different genetic group. And you can see that for most individuals, they are 100% like either yellow or green. You can see that we have here some very tiny portions of yellow, and here's very tiny portions of green. But basically, this is telling us that the populations we studied are genetically super different. We also analyze their morphology, and they're also morphologically distinct. So we measure a set of morphological traits associated with the sizes and relative lengths of their limbs and their tails. So this is showing that they are different, and also this figure shows like the general idea. So the lizards from the desert are smaller and have relatively longer tails and longer limbs compared to the others. So that's the morphology, and we also analyze their physiology, because if they are living in the desert, they need to adapt to these dry conditions. As we were just talking about, like how climatic changes can produce adaptation. So this happened in these lizards in the past. And what we see is that the lizards from the desert, here in yellow, lose less water than the lizards from the forests. So this plot is showing the distribution of the water loss for both populations. And here the line here in the middle represents like the mean or the median. So you can see that on average, the lizards from the deserts lose less water. So they're potentially adapted to their environment. And the last thing we analyzed was their signatures of adaptations in their genome. So we analyzed the different genes that were potentially under selection between these different populations. And we found in general signatures of adaptation in genes associated with water metabolism, their behavior, and their development. So just a few genes that were super interesting. So these genes that appear here are associated with the water metabolism, or the function and development of kidneys. And kidneys are very important for water retention. And also we found these genes that are associated with muscle and bone development. So that could also explain the differences in morphology that we detect in these lizards. Awesome. So to sum up, we have an evolutionary response of these populations to their different environments. And we have that the forest lizards have differences in their genome and in their physiology and their morphology when we compare them with desert lizards. So that's perfect. So if we go back to our original model, we have that these factors indeed are promoting the differentiation of lizard populations. And these are external factors. So the external environment is important. However, a cool thing that happens in nature is that even though populations can evolve under the same environmental context, they won't show the same patterns of diversification. So that's a big question. Like, why under the same context, some organisms don't diversify in the same extent? So just to explore this a little, like, let's go back to South America. So here we have this two group of lizards that have evolved in the Andes Mountains. And one of them is Leolemus, which is this radiation, and the other one are these Fimaturus lizards. And you can see in this photo, like Fimaturus, like even though they have differences in their colors, they all kind of look the same. Like they're all this chubby and flat lizard. But on the other hand, in Leolemus, we have different patterns of coloration and different designs. We also have differences in the scales. You can see like this guy has very pointy scales compared to the others. And we also have big differences in size. Like some of them are small, but others are really big and chunky. So that tells us that external factors are not the only thing explaining evolution. So what about the organismal factor and how those things affect evolution? And among those, we can have think of the genetic diversity of populations, their behavior, or even their ecology. And my research focuses a lot on how the genetic diversity affects the evolution of populations. So let's talk a little more about like genetic diversity and what do we study about genetic diversity. So genetics is the study of DNA, and DNA we know is like a sequence of different nucleotides. So if we have an hypothetical DNA sequence and we zoom in, you can see that we have all these nucleotides that have all the information that is contained in the DNA. But then, like across the DNA molecule, some regions of it correspond to genes. So some parts of the sequence will codify and generate proteins, and those are the genes. So, but it's only a small portion of the DNA. Like most of the DNA doesn't correspond to genes. On the other hand, the sequences flanking genes could correspond to regulatory regions. So these sequences here will determine the expression patterns of these genes, either the abundance or the timing or the location in which they are expressed. So these are two different components that are important, but we want to study the variation that affects these things. So one type of variation that I will show you today and that I'm interested about is the structural variation of these elements. So if you think of like our original sequence that is right here, what if it gets physically reorganized? So in this case, like this dark blue gene changed location with the light blue gene. Therefore, this regulatory sequence that is regulating the expression of this gene now is not regulating it anymore, but is regulating the other gene. So that will change and impact the evolution and the expression of those genes in the populations, because like the physical context of the gene has changed. The other type of variation that we are more familiar with are the punctual mutations. So let's say we have here our original sequence and there is a mutation represented by this red asterisk that could occur in a gene, so that could change the function of the gene, or mutations could also occur in the regulatory regions and hence change the regulation of the gene. So those are the two types of genetic variation that I will be showing you today. But I want to study and understand how this affects the evolution of lizards. So I will show you this other project in which we are exploring how the genomic variation promotes the evolution of anolis lizards. And I'm sure you are more familiar with anoles because they live here in North America as well. So what is an anole? So an anole is like this lizard that is characterized by having this dewlap. They also have toe pads, so they can climb better. And they have diversified extensively in the tropical Americas. So it's described as an adaptive radiation because we have over 400 species. So that's a lot of species. Um, and the cool thing about anoles is that they have very extensive variation in their phenotype and their morphology and their coloration and a lot of other traits. So we have big anoles, we have tiny anoles, we have anoles with big legs, with short legs. Some of them live in the crown of trees, other lives in the trunk, others on the ground. So they are very diverse. That's the message. So the question then is what generates such disparate traits within anoles? And for that, we focus on studying four species in anoles, and of which some of them have unique traits. So for instance, Anolis frenatus, this species here in this photo, is one of the largest species. And this graph is showing the distribution of body sizes across anoles. And you can see here in yellow that this is one of the largest species. And also you can see in this figure that is a very chunky lizard. And again, like this is studying lizards. Is large compared to other lizards, not large compared to an alligator. But it's big compared to the other anoles. We also studied Anolis auratus. This is one of the species that has the longest tail. And you can see in this picture that this tail is like disproportionately long. And Anolis carolinensis, the green anole. This is very common in Florida and Georgia and in that side of the country. And this is one of the species of anoles that lives in the coldest climates. So this is showing the minimum temperature of the coldest month, and you can see that carolinensis is the species that lives in coldest climates here. So here the question then is how the genomic variation of this species could be associated with their unique traits. So one of the first thing we studied was the structural variation. And here, if we go back to the idea of like each DNA sequence being like represented by a line, here is each block represents one chromosome or one DNA sequence. And you can see that they are physically and very similar. So you see that the first chromosome is red for all species, except for Anolis frenatus. So you can see that here we detected some really weird thing going on in the first 3 chromosomes of Anolis frenatus, that they are differently rearranged. Interestingly, we looked at the genes that were located here in the breakpoints between these chromosomes. So this basically means that chromosomes 1, 2, and 3 like broke and attached to other chromosomes. So it's like a physical rearrangement of them. So we look at which genes were located in the breakpoints, and we found genes associated with the development of their morphology. And again, anoles have a very diverse morphology, so that makes a lot of sense. And if we go back to how we understood structural variation, so if we change the physical location of genes, then that will impact how genes are regulated. So that's really cool. So maybe those structural variations are changing the regulation of genes within anoles. What else? So we also studied the punctual mutations, and we detected some very interesting things. So we detected mutations for Anolis carolinensis, mutations on genes associated with lipid metabolism. And again, carolinensis is a species that lives in very cold temperatures, and also with a higher temperature seasonality. So it is very cold in the winter, but not really cold in the summer. So besides being adapted to resist cold, it needs to be adapted to change its temperatures during the year. And we found signatures of selection in these 2 genes, the leptin and colipase, that are associated with lipid metabolism. So why is lipid metabolism important? So lizards eat insects, but they can store and they can use lipids as a way to store energy for the harsh seasons. So maybe they have adaptations in their metabolism to survive these periods that are harsher for them. So that's a cool thing we found with selection for Anolis carolinensis. For Anolis frenatus, which is this large species, and this plot is again showing the distribution of sizes for the different species, and you can see that this guy is disproportionately larger than the others. We found signatures of selection of genes associated with muscle growth. So maybe they are bigger because they can accumulate more muscle mass. And last but not least, when we studied Anolis auratus, which is the species with a really long tail, we measured the length of each one of the vertebrates in the tail. And we found that for auratus, that is here in blue, the vertebrates are longer than they were for the other species. And that is associated with, like, if you imagine how the vertebrates looks like, this is Anolis auratus, and this will be the other species. And we found mutations that, when have been tested in mice, produce the same outcome of an elongation of the vertebrae. So we have some mutations on specific genes, but that can impact a lot of the phenotype of these lizards. So just to sum up what we found with anoles. So anoles have dynamic genomes. So, and we saw that in their different structural rearrangement. And like, we also found other cool things that I'm not showing you today, but I will be happy to discuss more. And we also found signatures of selection on some genes with high impact on their traits, like such as the genes associated with lipid metabolism for cholinesterase or the muscle development for anolis frenatus. But now, like, okay, we understand these features of the evolution of this lizard. So why is that important? Because we're trying to understand the mechanisms that generate biodiversity. And we have this very diverse group of lizards that have very different forms. Like we have very tiny things, we have iguanas that are huge. So we need to understand what is generating this biodiversity, especially if we want to preserve it, right? And if we want to learn about their evolutionary trajectories, and how can that teach us as humans how to respond to environmental changes. So we know that they respond to environmental conditions, but they also respond differentially based on their unique traits. So when you have high or less genetic diversity, you will respond differently to the environmental changes. So how does this impact conservation? Like we live in a changing world, and you can see like this photo of this lizard like living in a urban environment. So it's adapting to novel conditions. One thing is that this kind of a study let us recognize the richness and diversity of evolutionary strategies that we have in lizards. So they have a lot of evolutionary potential, and they have done a lot of cool things throughout their evolutionary story. That is wonderful. And I think that's very important that we learn and appreciate how unique are their evolutionary patterns. And understanding these also help us to predict how they can respond to a changing world. Like if we are concerned about climate change or the habitat loss for this species, if we understand how they evolve or how they have evolved, we can then have a better idea of how they will respond to the future changes in their environmental conditions. And I think this is important because with this good biological knowledge, we can do informed management. Like we were just talking in the panel, like what can we do? Like we can maybe have a backyard that will sustain the lizard populations better, instead of, I don't know, like just destroying my backyard and not letting any lizards live there. Like we can propose strategies that will help them survive and thrive, even though the environment is changing. So I think with that, I will finish my presentation, and now I'll be happy to answer all of your questions or any comments that you may have. So thank you.